Salut. In this video, I will be showing you how to draw a eukaryotic cell, specifically a plant cell. So this is a generalized plant cell, and that means that not every single plant cell looks exactly like this, but we're drawing it so that we can remember the structures that are characteristic of a plant cell. I like to draw this as kind of a rectangle shape because it reminds me that plant cells are not squishy, they are rigid. And one reason that they're rigid is that they have a cell wall. So I drew one line, and I need to draw an additional line that's going to represent the thickness of my cell wall. Try to get this cell wall uniform in thickness. I don't have the steadiest of hands, so maybe you'll do better. All right. So we have this inner line representing our plasma membrane, and this structure here, the thickness here, this is representing the cell wall. Of course, we need a nucleus. You should be drawing in pencil. The only reason that I'm drawing in pen is that the document camera can't really pick up the pencil. So for diagrams and charts and drawings, you should use pencil. This is my nucleus. It's important to represent the nucleus as having a double membrane with these little holes called nuclear pores. Nuclear pores. So the nuclear pores allow messages to come from the nucleus and travel to the ribosomes, which are outside of the nucleus, so that protein synthesis can happen. We'll learn about that more later. But for now, you just need to understand that the nucleus has a double membrane and that there are these nuclear pores. And I'm going to add the nucleolus, or the nucleolus. That's the region where ribosomal RNA is synthesized. You can add it if you want to. Let's add some squigglies. I hope you realize what this is representing. That looks okay. And some more over here. Looks all right. Okay. We're going to add this kind of stack of flattened sacs. I hope you recognize that. And then adding this. So what have we done? These two shapes represent the endoplasmic reticulum. This stack of flattened sacs is the Golgi apparatus, or the Golgi complex. And these vesicles travel from the endoplasmic reticulum to the Golgi apparatus. And the contents of those vesicles are modified within the Golgi apparatus. And then often those vesicles are sent to the plasma membrane to expel something out of the cell. Because this is a plant cell, we definitely need a large central vacuole. Don't stress about the shape too much. The central vacuole is full of water, and it maintains the rigidity of a plant cell. So if you think about biting into some celery and you hear that crispy crunch, and you might squirt some water at somebody who's close to you, that crispy crunch is because of the rigidity or the hardness of the cell wall. And then you know that veggies and fruits are very juicy because they have this water in the central vacuole. And both of those things have the role of keeping those cells rigid. So plants don't have skeletons, obviously, like you do. They need a way to support themselves. So it's the central vacuole that's full of water and the cell wall. Let's add a lysosome here. I'm going to go over here and I'm going to draw a jelly bean shaped mitochondrion. It's important to know that mitochondria have a double membrane. So those folds are called cristae or cristae. It's important to represent those because mitochondria have an inner membrane and an outer membrane. And when we learn about cell respiration, it'll become more clear why that's critical. Since this is a plant cell, it's going to have a chloroplast, allowing it to perform photosynthesis. Chloroplasts are the same kind of beanish shape like a mitochondria. And we want to represent an inner membrane of the chloroplast. So chloroplasts have an inner membrane and an outer membrane. And the other thing that I'm going to represent, mostly to help you identify these on a micrograph, are these little stacks. So they're stacks of discs called thylakoids. An individual stack is called a granum. And they are connected to one another, so I'm drawing those connections. But the most essential thing to draw is the two membranes if you're doing this yourself. I'm missing something small but essential. Hope you realize what I'm drawing. So these are our 80S ribosomes. Okay, great. We might be happy with our drawing, but we definitely can't stop there because we don't have labels. So let us add some labels. This is the Golgi apparatus, or the Golgi complex, same thing. It's like the packaging center of the cell. This structure, I'm going to show it like this. This is the cell wall. So that entire thick portion is the cell wall. And then this inner line, just the line, is the plasma membrane, or the cell membrane, same thing. This is the central vacuole, full of water to keep the cell rigid. We have a lysosome, which is full of digestive enzymes. We have our good friend, the chloroplast. We have one mitochondrion. Let me label this vesicle. You could label any, obviously. We have the rough endoplasmic reticulum, or the rough ER. That's a standard abbreviation, that's fine to use. And then the smooth endoplasmic reticulum, or the smooth ER. Looks like I forgot my 80S ribosome. And when you make these label lines, make sure they're touching the structure that you're talking about. 80S ribosome. And then the jelly-like fluid that supports all these organelles is called the cytoplasm. Something I did not label. Take a look and see if you can identify the organelle that I did not label. See if you can notice it before I do. I forgot to label the very important nucleus.